Okay, good afternoon, everyone. It's nice to see you in this virtual world again. Please do me a quick favor. If you can hear me okay, please type yes into the chat window. Okay, wonderful, wonderful. Okay, so looking at your syllabus, we are 2 weeks away from our second lecture exam. And so what I will do is, if everyone in the class joins us today at some point, I will give you the study guide for your lecture exam 2 today. If it looks like Hayden hasn't joined us yet, so perhaps she has something else going on. If not everyone is able to join us today, then what I will do is write up a study guide for you with some hints about what will be on lecture exam 2 in 2 weeks. So I noticed that some of you have already created that free account at the Classmarker website. As I had indicated with my previous email to you, PAC has decided to keep its campuses closed for the remainder of the semester. So it's unfortunate, but it is in the best interest of public safety. So folks, we're going to know each other real well through the computer, but we're not probably going to meet face-to-face again. So we'll be giving lectures as best we can online, taking exams online, doing office hours online. So hang in there, send me emails, let me know what you need to succeed in the class. Let me know how I can help you. Before we start new content today, do you have any questions? Okay, I'm not seeing any questions. So folks, we're going to get started with chapter 9. So the topic of chapter 9 is the genetic code and transcription. So let me start sharing my computer screen and bring up a Word document so we can take some notes. Okay. And let's see. No. Just one moment, please. Okay, gang. So at this time, I think you should be able to type in messages in the chat window if you have a question or you need me to slow down. At the same time, please feel free to unmute your microphone if you want to talk, if you want to ask a question. That's quite okay. All right, so these are going to be chapter 9 notes. And today's date is the 26th. Okay, so here we go, gang. Hang in there. I know this is not an optimal circumstance. Okay, so let me give you a quick comments about foundational information. So just some talking points that you should keep in mind while we're having our discussion today. First off, you know that when DNA is being used to make RNA, that process is called transcription. And I know not too many of you yet have had microbiology, and it's probably in a microbiology course you would get exposed to the next idea, but you can appreciate that in the central dogma where we have DNA going to RNA and RNA going to protein, part of that flow chart is reversible, and it's the transcription process that is reversible. There are circumstances in Mother Nature where RNA can be used to make DNA, and in such a case, that process is called reverse transcription. Okay. An example of reverse transcription would be a virus like HIV, the human immunodeficiency virus. It's an RNA-based virus that is capable of performing reverse transcription. Okay. All right. So please stop me if you can't hear me. If there's a problem, please stop me so I can correct that for you. Okay, now you notice the title of this chapter in your textbook is the genetic code and transcription. Okay, and that's important that you appreciate. You know, you probably remember from Bio 101 and 102 that you have these things called codons in the messenger RNA that are used by a ribosome to translate and make protein. Okay, those codons are the genetic code, and those codons are always RNA based, they're not DNA based. In other words, from a historical perspective in the development of the field of genetics, the phrase genetic code refers to the information content of the messenger RNA, not the DNA. Okay. So please understand that the phrase genetic code is defined from the perspective of a ribosome decoding the information of the messenger RNA. Okay. You're going to find, gang, that over the next couple lectures, I swap back and forth between topics of chapter 9 and chapter 10 because it's very natural to do so, right? I mean, when we talk about transcription, the next logical process after that is translation. So you may find that I go back and forth a little bit between ideas in both chapter 9 and chapter 10. And just so I know I put it in your notes, we know that the process of translation, which is using the genetic code information in the RNA, is what's used to manufacture protein. Protein is manufactured by having a ribosome interpret the genetic code, okay, in a process called translation. Okay, so hopefully no mysteries here, and by all means interrupt me or type a question into the chat window if you have it. Okay, keep in mind that on our planet, there are two major categories of cells, prokaryotic cells and eukaryotic cells. And it's important that you appreciate that in prokaryotic cells, transcription and translation are coupled. They can happen at the same moment in time because prokaryotes do not have a nucleus. Remember that in prokaryotes, transcription and translation is coupled. This means that transcription and translation can happen simultaneously because prokaryotes, and when I say prokaryotes, that's almost synonymous with bacteria, because prokaryotes, bacteria, do not have a nucleus. Okay. And of course, just to be, anal about it, we'll mention that in eukaryotes, transcription and translation are uncoupled. Don't know why I want to type bad today. In eukaryotes, transcription and translation are uncoupled. This is because eukaryotic cells do in fact have a nucleus. Okay, so I'm just going to take a moment to breathe, because I'm hoping that I'm not typing too fast for you. I know that you're trying to keep up at this time. Okay, last thing, actually not the last thing, number 6, we are coming off our lecture and discussion of DNA replication in Chapter 7. And while it's probably obvious, I want to be sure that you truly get this, that when we talk about DNA replication, remember, we're talking about copying the genome, making a copy. That is a different process than expressing or decoding the information in the DNA. So what I'm saying is DNA replication is one process, which is copying the information, whereas transcription and translation are expressing or using the information, decoding the information. Okay? DNA replication is copying the genome, while transcription and translation are using, decoding, expressing the information of the genome. Okay? I mean, that kind of seems true at face value, but the reality of it is when we start talking about questions in homework and we start getting challenged by questions on exams, sometimes these basic principles kind of float out the head a little bit. So just be careful that you appreciate these things. Okay, number 7. All right, we're going to be going fairly meticulously through the process of transcription in bacterial cells, prokaryotic cells, and again, at the end of that, contrasting with some differences in eukaryotic systems. But let me simply mention here that RNA polymerase is the major enzyme that performs transcription, and that is true for both prokaryotic and eukaryotic cells. Okay, RNA polymerase is the major enzyme responsible for performing transcription. Okay, and keeping in line with some of the information that was shared last time, actually, because we talked about DNA replication requiring an RNA primer to jumpstart the DNA replication process. Consistent with that is that RNA polymerase does not require its own primer to begin its work. It doesn't require that. Only DNA polymerase requires a primer. Okay? Unlike DNA polymerase, RNA polymerase does not require a primer to start its work. Okay? RNA polymerase does not require a primer to start its work. Like DNA polymerase, RNA polymerase also synthesizes new RNA, you guessed it, in a 5' to 3' direction. RNA polymerase synthesizes new RNA in a 5' to 3' direction. Okay, that's a take-home message that's very important, gang. I know I keep saying it, and you're probably sick of me saying it, but as far as we have ever encountered on planet Earth, as far as any systems that scientists have ever studied, DNA polymerases and RNA polymerases always synthesize their nucleic acid in a 5' to 3' direction. It does not happen the other way as far as we know. Okay. Oh, it'd be much nicer if I was in front of you. I could actually see you, but unfortunately, that's not so possible right now. Okay. Okay, let me see. What else? A couple other talking points here. You remember that when we were talking about DNA replication in Chapter 7, we needed a separate enzyme to unzip the DNA helix, and the name of that enzyme was helicase. Well, during transcription, the DNA helix is also going to need to be unzipped, but we don't need a separate enzyme to do the unzipping. There is no helicase in transcription because RNA polymerase is capable of doing double duty. RNA polymerase can simultaneously unzip the DNA helix and synthesize the RNA. Okay, so Mother Nature endowed this enzyme with some additional multitasking capability, if you will. RNA polymerase is capable of both A, unzipping the helix, unzipping the DNA helix, and B, manufacturing new RNA. Okay, so I'm trying to highlight for you some of the subtle but important differences between DNA synthesis and the manufacture of RNA transcription. Okay, lots more to do here. Let me pause and ask you, do you have any questions at this moment in time? Okay, not seeing anybody passing out yet. I guess that's a good sign. So, what I'm going to do next, folks, is we need to get into the nitty-gritty of the transcription process, but I think it would probably be good to show a basic overview cartoon and overview video about this process. And expect in the email that I send you later today, or hopefully by the end of today, I will include a link to the video I'm about to show you, and I may also copy some additional links to some additional videos that I think are particularly good at explaining some of these processes. So that will be coming in your email later today. But okay, let me show you a short 3-minute video on the process of transcription in prokaryotes. So bear with me just a moment while I get that queued up. Okay, so at this point you should be seeing my screen, and I'm at YouTube, and I'm showing a video that's called Transcription High Definition Animation by McGraw-Hill, and I will send you a link for this later today. So let's listen to this. It's about 3 minutes long. Here we go, gang.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene. In bacterial cells, the transcription of a gene...
Okay, folks. Sorry about that annoying little pop-up there. Some of the videos on YouTube are not always the greatest, but we did get through the primary content of the video. Just a quick check. Are you folks still with me? Please type a yes in the chat window. Okay, thank you. I just want to be sure that I'm not losing you. Okay, so the process that the video just tried to explain, obviously, there's a lot there that they crunched into a 3-minute time frame, but it is a nice summary of the process, and we need to step through that process together here in our notes. And so I'm hoping to hit most of the highlights and some other things, in fact, that the video didn't talk about. But since you've just seen the video, do you have anything you'd like to ask? Any questions before I start talking about the process?
So I do have a question. So this video obviously demonstrated prokaryotic. How would that differ in eukaryotes?
Oh boy. Yeah. Let me defer your question right now, Kevin, because that is a talking point that I'm going to do after we get here. There are a considerable number of differences. Let me just say about 2 of them. Prokaryotic messenger RNAs don't have a structure that eukaryotic messenger RNAs have on their 5' end called a cap sequence. It's actually a chemical called 7-methylguanosine. So that's a difference. Another difference is that most eukaryotic RNAs that get made, they have what's called a poly A tail on their 3' end, And you don't find that as commonly in prokaryotic transcripts that are manufactured. So that's just two of a whole toolbox full of things that are in fact different between prokaryotic and eukaryotic transcription. So maybe not today, I'm not sure how far I'm going to get, but certainly by the end of next class, when I'm talking about eukaryotic differences, if there's something that's particularly on your mind, please bring that up again. Okay. Okay. Folks, let's talk about transcription here. Okay. Transcription in prokaryotes. Okay. All right. So, first talking point. You know, I think you already know that RNA polymerase recognizes the start of a gene by recognizing the promoter. Okay. RNA polymerase knows where to begin transcription by identifying the start of a gene. The start of a gene is the promoter. Okay, the start of a gene is a promoter. And you recall, I think you recall, you know, some commonalities here that we said that, you know, question to everyone, and I'd ask you to please type your answer into the chat window. Is the promoter a sequence of DNA or a sequence of RNA? Okay, all right, good, fantastic. Okay, so everybody's got that, right? A promoter is a DNA word. It makes sense that it has to be, right? Because you got to identify it before you even start the process of transcription. Okay, remember that a promoter is a DNA word. Okay. Next talking point. Is the promoter actively synthesized into a sequence of RNA? Go ahead and take a shot at answering that in the chat window. Is the promoter actually transcribed into RNA? Okay. All right. Got some interesting comments there that everybody can see. Sean and Caitlin said no. John said yes, but it's removed. The answer is no. The promoter is not synthesized into RNA at all. But thanks, John, for trying. Okay. The promoter DNA word is itself not transcribed into RNA. The promoter is simply a binding target for RNA polymerase. Okay. All right. And last thing that may come out later in our conversation, I'll just put it down here in your notes, is that you appreciate most promoters in bacterial cells, bacterial systems, most of these promoters that we've cataloged through statistical analysis, you know, with the computer jocks that look at all this stuff with the DNA sequences, they tell us that on average, most prokaryotic promoters are roughly 40 base pairs upstream of the transcription start site. Okay, so 40 nucleotide pairs of DNA, base pairs, upstream of the transcription start site. So you appreciate what we're going to get into talking about gene architecture this hour together. Okay, but the idea here is that, you know, we might have some sequence of DNA that would be, I don't know, we'll say minus 40 nucleotides, and it would be some random sequence of DNA, minus 40 nucleotides, and it would continue along till we get to minus 1, and then there is no 0. It goes from minus 1 to plus 1. The plus 1 indicates the start of transcription. Okay. So everything that is not transcribed, you notice, has a minus sign to indicate its position. Everything that is transcribed has a positive sign for the number. Okay, let's continue. RNA, okay. It's easy for scientists, including me, to slip into common assumptions, and we know that assumptions are dangerous. But let me say this, that E. coli, which is a typical bacterial cell that we know quite a lot about because we've been studying it for decades, E. coli bacteria, its RNA polymerase is a quaternary protein made of 5 polypeptides. Okay, E. coli RNA polymerase is made of 5 polypeptide units. They are alpha, beta, beta prime, omega, and sigma. Okay, so I'm going to give you a very quick description of what these polypeptides that are part of the bigger enzyme. If you've studied any enzyme biology in other courses, you know a structure like this is going to be called a holoenzyme. But at any rate, let's talk about the subunits. The alpha subunit of RNA polymerase is a polypeptide within the larger enzyme that provides a contact surface for other regulatory proteins that want to modify the transcription process to attach to. So think of alpha as a bridging subunit to other regulatory proteins that want to modify the transcription process. Provides a contact surface, means it acts like a bridge for other regulatory proteins in the cell to bind RNA polymerase and alter the transcription process. Okay. All right, we have, I'm going to put beta and beta prime together. Beta and beta prime polypeptides are the core of RNA polymerase's enzymatic activity for actually synthesizing new RNA. Beta and beta prime are the core subunits of RNA polymerase that are responsible for the polymerization of new RNA. Then we have omega, the omega subunit is a protein that helps to assemble the entire RNA polymerase complex. You might think of it at a construction site. You have the worker bees that are doing their projects at the construction site, but there's always a foreman that helps ensure that everybody knows what their job is and help keeps the process organized. That's sort of like what the omega subunit is all about. the omega subunit facilitates the assembly of the entire RNA polymerase enzyme. And apparently through structural studies that have been done with crystallographers that study these sorts of things, the omega subunit also seems to be involved in once all the subunits have come together, the omega subunit kind of helps them lock into position and stabilize the overall structure of the enzyme. Okay, the fourth note on these 5 subunits is the sigma subunit. And the sigma subunit helps RNA polymerase find, recognize, and attach to the promoter. Okay. One question that hopefully is coming up in your mind, or at some point has come up in your mind, is, well, if all it takes to express a gene is for RNA polymerase to sit down on a promoter, it kind of seems like every gene in the cell should be expressed simultaneously all the time. And obviously that's not true. Well, from some of the things we've talked about in this course that the structure of the chromatin plays a huge role in whether a gene can be turned on or turned off. But at least in the case here of prokaryotic transcription that we're talking about, some of the gene regulation, some of the ability for the cell to selectively turn genes on or off has to do with controlling the binding of RNA polymerase to the promoter. And that selectivity is achieved by having different versions of sigma. E. coli and other bacterial cells have multiple different versions of sigma. Different versions of sigma help RNA polymerase find different categories of promoter. Okay. Different sigma factors exist, which helps the bacterial cell control which promoters are bound at what moment in time to regulate the process of gene expression. What we're talking about right now is transcription, right? Gene expression involves typically 2 things, transcription and translation, but we're talking about transcription right now. Okay. All right. Questions, gang? Excuse me, I'm just looking at my notes and I'll pull down in just a second here. Okay. All right, any questions at this time? Okay. As much as I like your textbook, of course, there's no resource that's perfect. And honestly, I think this chapter is a little weak on graphics and cartoons. I mean, basically the only cartoon that your textbook gives you about bacterial transcription is figure 9 on page 250 of the current edition of the book. And so, you could, maybe have that open in the surrounding pages as you look at things. If you go on to more sophisticated courses in the upper levels of your undergraduate education or in graduate school, you'll get textbooks that have, deeper illustration of these topics. Particularly molecular biology and biochemistry textbooks typically have deeper illustration of these things. So that's why I want you to rely on the notes I give you, the videos I'm going to share with you, your homework to try to get a collective feel of how this process actually works. Okay. Number 2. point number 2, okay? Let's see here. Binding of RNA polymerase to the promoter represents one way that transcription can be related. And that I've sort of hinted at that with Roman numeral, the little Roman numeral I up there. I probably should have made that a little a. Okay, so let me just put that in your notes. Binding of RNA polymerase to the promoter represents one way transcription can be regulated. Okay. Let's take a look at that a little bit deeper here, okay? Let's talk about bacterial promoters. Okay, bacterial promoters have a consensus sequence. What I mean by consensus is, if you look at the DNA sequence of like 100 genes and you compare the DNA sequence position for position across the 100 molecules of DNA, you will find some areas that are frequently very similar even between genes. I may sneeze, please excuse me. Okay, all right. So bacterial promoters have a consensus sequence that is hallmarked. It's good I'm not sneezing in front of you, isn't it? Although I don't think I have coronavirus. Okay. Bacterial promoters have a consensus sequence that is hallmarked by 2 stretches of 6 nucleotides each. Okay. And so what I can say about this is that the first sequence is, okay, please hear me on this. The first sequence is often confused with the eukaryotic Tata box. It is not the eukaryotic Tata box because we are talking about a bacterial cell at this time. But the sequence is this: 5 prime, TATAAT, 3 prime. Okay, this is commonly found at the -10 region of the upstream of the transcription start site. It's found at the -10 position of the gene. Okay. This sequence, which you could, you can see why people confuse this with the Tata box of a eukaryotic promoter, because obviously it has TATA, Tata. You can see why it would be confused, but this sequence in prokaryotic biology is actually known as the Pribnow box. Okay. All right. Okay, so that's, that sequence, 5 prime, TATAAT, 3 prime, that sequence is one of these 6 nucleotide stretches that represents a consensus sequence that you find it very commonly upstream of prokaryotic genes. The second sequence is this one, 5 prime, TTGACA, 3 prime. This is commonly found around the -35 position of the gene. Okay. Okay. So I'm going to give you a few more notes here, 1 or 2 more statements about this, and then I'm going to switch gears and go over to a drawing that I made before class to talk about this, and I'll present it to you so that you can copy it down for yourselves. Okay. Letter B here under number 2. Okay, so these consensus sequences, the Pribnal box and the TTGACA box, if you will, okay, if there are mutations in these hexamers, if there are mutations in the DNA sequence of these consensus sequences, there's typically a dramatic reduction in transcriptional output from the gene. So mutations in the -10 or -35 regions of the promoter reduce transcriptional output from the gene. Quick little story, gang. When I was in grad school, one of the first projects that my supervisor gave me was to clone a gene in E. coli called NUS-A. NUS is short for N utilization substance A. No big deal, who cares? But so I spent a few months cloning this gene, and I got to a point where I had the gene cloned, and the bacterial cells that the gene was cloned in was in fact taking the gene and making the protein that was encoded from the instructions. I had evidence of that. I could measure it in the lab. Everything looked good. So I took this strain of bacteria that I had genetically modified, and I stored it in a deep freezer at -80 degrees Celsius. About a year later, I wanted to use this strain of bacteria again, so I got that strain of bacteria out of the -80 freezer, and I started doing new experiments with it. And lo and behold, when I got it out of the freezer, the bacteria stopped making this protein from the clone gene that I had generated a year before. Well, after a lot of cursing, I finally figured out through sequencing of the bacterial DNA that while the bacteria was sitting at -80 degrees Celsius in this deep freezer, the promoter region of the gene that I had cloned had mutated. And it was in the area of the -10 and -35 hexamers. Long story short, that I had made a genetically modified bacteria produce a protein that apparently it was not happy to produce. And so while it was sitting in the deep freezer, even at -80 degrees Celsius, it still mutated its DNA to turn the expression of that gene off. So these are the kind of weird things that happen in the real world of science. And, you know, cells that you're working with don't necessarily cooperate with you simply because you're trying to become a scientist. Okay. So, letter C. Variation in the nucleotide sequence along the entire promoter can be intentionally used by genetic engineers to increase or decrease the amount of transcription from a specific gene. We now have the understanding that technology, we're sophisticated enough that we know exactly what mutations to put into that region of the promoter to increase transcriptional output as well as decrease transcriptional output. Variation in, okay, so I'm repeating myself. Variation in the nucleotide sequence along the promoter can be used to up or down regulate output from the gene. Okay, gang, before I continue any further, do you have any questions? Okay, I'm not seeing anything from you, so I hope you're not passed out on your living room floor. Okay, so let's go to a challenge that I have for you here. I'm going to go to PowerPoint. Okay, And so here we have a little sketch that I've drawn. And folks, I want to encourage you to please draw this sketch in your notes. Excuse me, I got a telephone call. Okay, so please sketch this sequence out in your notes and write down these questions. I'll give you a couple minutes to do that. Okay. So as you complete sketching this out in your notes and writing these questions down, go ahead and see if you can answer any of these questions. Okay, folks, I'm going to resize the window so hopefully I can get the chat window on a little bit. Bear with me here. Okay. If you have completed writing these notes out, or when you do complete it, if you would please type in completed or done in the chat window so I know everybody's ready to talk. Okay, gang, let me see here. All right, thanks everybody. So let's take a look at that first question. Which strand of DNA contains the gene? So up here, where I'm circling with my cursor right now, I've written a top and bottoms. That's how we can tell these two apart, the top strand or the bottom strand. Which strand of DNA contains the gene? What would you say? Go ahead and either, if you want to talk, unmute your mic and say it, or if you want to answer it in the window, go ahead. Is it the top strand that contains the gene, or is it the bottom strand that contains the gene? Okay. Just trying to give everybody a chance to answer here. Okay, so we do have some top answers and we do have some bottom answers. Okay, all right. So the answer, in fact, is the bottom. The bottom strand contains the gene. Okay, so first let's write that down. I'll try to write that down in a way that you can actually see it here. Okay. Okay. All right. If you notice, and I'm sorry I don't have, I'm not in front of you and it's hard to highlight things, but if you notice here, this sequence here that I'm going back and forth with my cursor, that's going to be your messenger RNA, right? Because that's the product of transcription. You notice that this sequence, UCAUU, AAC, is the same thing as it is up here, except for an RNA you have U's, whereas DNA you have T's, right? So U is the same thing as T, DNA to RNA, C, C, A, A, T, T, U, U, A, 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 A. Okay, so this messenger RNA sequence is the same as this, which means the bottom sequence was the template to actually make the RNA. It's the template sequence, which is the one that contains the gene. Okay. So let me highlight that this is the template sequence. The template sequence always contains the gene. Okay, so please don't think that in every drawing of a gene that the template sequence is always going to be on the bottom of the drawing. That doesn't have to be true at all. I could definitely have flipped this on you. So please don't think that the template sequence is always the bottom strand of a DNA helix that's drawn left to right on a piece of paper. That is not true. Okay, so what you have to do is figure out, well, what sequence of DNA could have been used as a template to actually make the messenger RNA. The sequence of DNA that you isolate that is the template to make the RNA, that's where your gene is. Okay? Does that make sense, folks? Any questions? Okay. So how about the second question? What is the chemical directionality of the top strand? Okay, so up here on the top strand where I'm circling with a T, should I have a 3 or a 5 prime there? Should I have a 3 or a 5 prime? Okay, so Kevin, you say 3 to 5 prime, but you wrote the word bottom. What do you mean by that? So,
That was my previous response to the last question.
oh, okay. Thank you, Kevin. Appreciate it.
I think it all just lumped into one. Sorry.
Okay. Yeah, yes, the chat window's doing that. Okay. 3 prime to 5 prime, right? So, exactly. Here's the messenger RNA. I had already given you a clue that this is the 3 prime end, and this is going to be identical, which means that that would be, in fact, 3 prime up there. And 5 prime here. Okay. Now, gang, look back in your notes on your Word document. Did I draw the TATA? Did I draw the TATA-AT sequence, the -10 hexamer? Did I draw it correctly from 5 prime to 3 prime? Did I draw the I don't want to say TATA box because that'll confuse you. Did I draw the -10 hexamer correctly? Hopefully you see that I did, right? 5 prime to 3 prime. If you read it, you don't read it left to right across the page. You always read it according to the chemical directionality of the sequence. Always read DNA and RNA according to the chemical directionality. If you read according to the chemical directionality, it's TAT, AAT, so that is in fact correct. Okay, so just to finish the note taking here, the chemical directionality of the top strand would in fact be 3 prime on the left side and 5 prime on the right side. Okay, so question number 3, did RNA polymerase start its work by attaching to the left or the right of the bottom strand? So in other words, did RNA polymerase start here, where I'm circling right now, or did it start over here? Did it start to the left or to the right? Okay, Jose says to the left. John says to the right. Exactly. Remember, these sequences here are in fact binding sites for the polymerase. So that means that the RNA polymerase started here. And remember that there are going to be sequences of DNA here that have an exposed on the bottom strand, there's going to be an exposed 3 prime end so that the 5 prime end of an incoming nucleotide can link it so that this 5 prime is to this 3 prime. Okay? So yes, RNA polymerase bound to the right side of the bottom strand where the actual promoter elements are. Okay? So in other words, this bottom strand, okay, this bottom strand, when it was used as a template, RNA polymerase had to be there at the promoter elements. Okay, last question. Question 4. In gene or bioinformatic databases that report the CDS, the coding DNA sequence. So C is coding DNA sequence. Would their top or bottom strand be reported? And honestly, I know you have no background for this, so I'll simply tell you, because we define the bottom strand as the template sequence, by definition, that means that the top strand is the coding sequence. This is the coding DNA sequence. Okay. Now, the answer to question 4 is in fact the top strand, because DNA, gene, bioinformatic databases always record their DNA as the coding sequence. It's just a convention that's used in the community of scientists. Could it be done the other way? Yes, it could, but it's by convention that gene and bioinformatic databases always use the coding sequence. Coding, in this case, is the top strand, okay? In this case, all right? So, the reason, if you're curious about this, and this is something I'm going to tickle you with in a future homework assignment, but at any rate, the reason that the coding sequence is the one that's stored in the database is because if you look at it, the coding sequence is the DNA version of the RNA, right? The coding sequence is the DNA version of the RNA. Didn't we say that? T is equivalent to U, C is C, A and A. The T's are equivalent to the U's, A and A and A. By recording the coding sequence of the DNA, then if you want to ask the computer to quickly translate it, all you have to do is translate the DNA version of the RNA. You don't have to retranscribe it. So it's computationally more efficient to store the information in databases this way. Okay, it's computationally more efficient to store CDS sequences. Okay, gang, are you okay right now? Please let me know if I've confused you. Let me know if there's something you want to talk about or ask about. Do you have a question? What's on your mind right now? Is there anything you need to talk about? Are you passed out on the floor? Okay, all right. I appreciate hearing that because sometimes I don't know what I need to do with you to help you understand something. And that's why you get it, I'm a little old school. I really prefer writing things down, drawing and sketching than I do watching videos and typing on a screen. That is not the way to learn biology. Okay, all right, folks. So if there's no other questions about this, we'll go back to some basic note taking. Keep this note right here is important. It's not important simply because you got to know it to understand genetics and biology. Some topics about coding sequences and template sequences could show up in your next exam. Hint, hint, hint. Okay, let's go back to the Word document. Here we go. Okay, I know we're getting short on time here. Okay. Let's see. Number 3. Okay, so we're talking about the process of transcription. I've been giving you some comments about RNA polymerase, about the architecture of a gene. Okay, so let's assume that RNA polymerase has identified its promoter and it has attached to the promoter. So following RNA polymerase binding to the promoter, the enzyme unzips, and you've heard me use this word melt, M-E-L-T. It melts a small region of the DNA, about 12 to 14 base pairs worth, and begins polymerizing, adding one RNA nucleotide to the next in that 5' to 3' direction. That's the initiation of transcription. Okay, following binding of RNA polymerase to the promoter, the enzyme melts, okay, meaning unzips a small region of double-stranded DNA, about 12 to 14 base pairs worth, and begins adding, meaning polymerizing, RNA, 5' to 3', okay, where the 5' end of the incoming nucleotide links to the 3' hydroxyl of the growing chain, okay. This is initiation, transcription initiation. Okay. Moving on, simply because I really want to get some notes into you, gang. Number 4. After the first 12 to 14 base pairs of new RNA have been synthesized, the sigma factor, the sigma subunit of RNA polymerase, detaches and falls off the core enzyme. The sigma factor of RNA polymerase falls off the promoter and detaches from RNA polymerase. Okay. At that point, sigma factor has fallen away, and the rest of the core enzyme continues to make RNA. Okay, I'm going to start typing RNAP to indicate RNA polymerase. The rest of the core RNA polymerase enzyme continues to make new RNA, and this is elongation. Okay, and as I believe I mentioned, perhaps to maybe one of Kevin's questions last class period, when we're talking about polymerase molecules like DNA polymerase and RNA polymerase molecules, the general rule of thumb is that most bacterial polymerases will polymerize at a rate of about 40 to 50 nucleotides per second at a temperature of 37 degrees Celsius. E. coli RNAP makes new DNA, makes new RNA at a rate of 40 to 50 nucleotides per second at 37 degrees Celsius. Okay, I see it's 1:46, gang. So I guess we're going to have to pause. Okay, I'm going to pause just for a moment to let you finish your note taking here. Okay, so go ahead and finish your note taking. Please tell me when you're done taking your notes. Let's see here. Okay. Caitlin, are you okay? You still working on those notes? Okay, Caitlin and Jose done. Fantastic. Let me stop screen sharing here so I can talk to you. Gang, I'm going to start an office hour in 15 minutes. And I really want to encourage you to stick around because There's just so much meaty content that we've got to cover to get you ready for your exam in 2 weeks. So in 15 minutes, I'm going to review homework problem number 3 from chapter 5, which most of you had a little bit of problem with. And I'm also going to talk a little bit about DNA recombination from chapter 7. It's a topic that actually Hayden asked me to talk about, and unfortunately she's not here, but I will record it on the video and everybody can get some benefit from it. Recombination is an important big topic. Unfortunately, it's also considerably complicated, so we need some time together to go over this. So if you really can't stay, I understand, but I want to encourage you to stay. And if you have a pack of colored pencils or pens nearby, that would be very useful to you. So if you're going to stay, let's meet up at 2:05. I'll be back online with you at the same Zoom channel here in 15 minutes. See you soon, gang. Okay, gang, welcome back, and thanks for sticking around for, maybe another 30 to 45 minutes of genetics fun. So, okay, so Ian, could you give me a thumbs up in your video screen if you can hear me? Thank you. Okay. All right, gang. So I wanted to cover one of your homework problems from chapter 5, because part of the problem, the first part of the problem, most of you did okay with, but the second part of the problem, you folks kind of jumped to a wild conclusion about something. And I want to be sure that we cover this kind of thing. So if you have chapter 5 open, go back to the homework problems. It's number 3 that we need to talk about. Let me read it here while you maybe find it in your textbook. It's on page 139 of your current textbook. Okay, number 3 says, streptomycin resistance in Chlamydomonas may result from a mutation in either a chloroplast gene or a nuclear gene. First question, what phenotypic results would occur in a cross between a member of a mating type positive strain resistant in both genes and a member of a strain sensitive to the antibiotic? Second question, what results would occur in the reciprocal cross? Okay, so it was the reciprocal cross that I think most of you had a little bit of problem with. So let's take each of these at the same time. Okay, so I'm going to share my screen again and go back to a PowerPoint presentation. Okay. All right, so that's what we were working on at the end of class. Okay, here we go. Here's chapter 5, question 3. On the left side of this vertical line, I've sketched out the scenario that we're dealing with for the very first question. And on the right side of the page, I've sketched out the start of what would be the answer to the second question. So go ahead and diagram this out in your notebook someplace. And when I get a indication that you're done in the chat window, we'll talk about this. Okay, just a second while I manage my windows here. Okay. Okay, go ahead, gang. Please diagram out what you see in front of you. Okay, I'll use some words here if you can't quite read my writing. What you have up here is mating type positive. Mating type superscript plus is mating type positive. And from the problem, we know that we have a nuclear, NUC, nuclear resistance gene, and we have a cytoplasmic or chloroplast resistance gene, a gene that provides resistance to streptomycin. We're going to cross that with a mating type minus that is sensitive. So that means in this scenario, as it was defined by the problem, we have a nuclear sensitive scenario and a chloroplast cytoplasm sensitive gene. So that's my cryptic writing there. Please let me know when you're finished copying the notes. Okay. I got your message, John and Ian. We'll just wait on everybody else. Okay, Kevin. Okay. Okay. All right. I think that's about everybody, I think. Okay. So let's walk through this, gang. Okay, so again, to the left side of the vertical line, we're talking about the first question. The first question is, what phenotypic results would occur in a cross between a member of a mating type positive strain resistant in both genes, that's why we have the resistance symbols right here, and a cross with a member that is sensitive to the antibiotic. Okay, so that's why we have the S here for sensitive in the mating type minus. Okay, so first off, most of interpreting this problem, you would had to have read the information on pages 125 and 126 of your textbook very carefully. And while I'm not going to type that out for you, let me make a few statements that I think you probably have already read and are aware of. So to do this problem, you have to appreciate that number one, this is an algal cell, right? This is an algae. This algal cell is a haploid, okay? So we're saying N equals 1 is a haploid state. We know that when the two cells that are haploid fertilize, we get a diploid zygote, okay? We know from the research that's been done that both the positive mating type and the negative mating type contribute equal cytoplasmic contributions to the zygote. So we are not dealing with a maternal effect kind of scenario here. We know that the streptomycin resistance phenotype is only transmitted through the positive mating type. How do we know that? Well, because the textbook tells us that. How does the textbook know? Because scientists have done experiments with this to determine this. So we know that the resistance phenotype always is transmitted through the positive mating type. The last piece of information is this. Okay, so if we have these gametes fertilize and become a zygote, we have a zygote with the combined genes here. So we have a resistance gene in the nucleus and a sensitive gene in the nucleus. We have a resistance gene in the chloroplast. We have a sensitive gene in the chloroplast. And as you may remember, because I think most of you did this problem correctly, the key piece of information to realize is that when the zygote forms, apparently the genetic information from the negative mating type chloroplast degrades. Okay, so this genetic information in the chloroplast actually breaks down. So I'm going to take a red pen here and I'm going to take a red pen and I'm going to put a little X through that chloroplast gene, because you can see that the sensitive chloroplast gene came from the mating type minus. Okay, now exactly why that happens or how that happens, the actual mechanical process of how that happens, we don't know. We don't understand it. It's still an active area of research for people that study fungi. But what we do know is that the mating type gene in the chloroplast degrades. So the MT minus gene, or you could simply say the mating type minus chloroplast DNA, degrades. Again, why it does so, we truly don't know, and I can't explain that to you. So this is the situation that happens then, okay? So we have the fertilization that occurs. We have meiosis continue again to produce haploid cells, right? We're going to have haploid cells back here again. And the possible results of the meiotic process are in fact going to be one kind of cell that has a resistance gene in the nucleus and a resistance gene in the chloroplast. Why isn't there any sensitive gene in the chloroplast? Because again, it degraded up here. The other possibility, when you split the genetic cargo in half, you're going to get exactly the same scenario where you have a resistance gene in the nucleus and a resistance gene in the chloroplast, meaning that all progeny are resistant to streptomycin. Okay, I've already answered my question over here of why. The why is there is no sensitive gene in the chloroplast because it degrades. Okay, it degrades. Okay, that, folks, most of you did in fact get correct, and I was happy about that. Before I go any further, do you have any questions, anything you'd like to type in the chat window?
So I have a question here. Regarding the sensitivity of the nuclear gene, what sort of influence does that have? Or do we expect that to be degraded also or exclusively in the
Kevin, thank you for saying that, because actually I have a typo on my drawing. And I appreciate you saying that. I have it right in my notes and I drew it out here in the diagram incorrectly. That second possibility here should in fact be a sensitive. So thank you for saying that. Let's fix that right now. So that would be sensitive. Okay, that makes things a little bit better. You see that the nuclear genes partition. You get one with a resistance gene and one with a sensitive gene. But it turns out that both cells, both haploid cells, their only source of chloroplast DNA comes from the positive mating type, which you can see here at the top contains the resistance gene. So that means both of the cells produced down here at the bottom are going to in fact have the resistance gene, and you only need one copy of the resistance gene in either the chloroplast or the nucleus to in fact get the resistant phenotype. Okay, so yes, that's better. Thanks, Kevin. Any other questions?
So what determines if you're a positive mating type or a negative mating type? Is that your resistance or your sensitivity? So only algae that are sensitive, their genes like degrade, and that's the case? Or is there some other determining factor?
There is some other determining factor, John. Without wanting to go into the deep genetics of this, you can kind of think as mating type positive as male and mating type negative as female. And what I'm saying here is that there are specific genes on specific chromosomes in these algal cells which will define the mating type for the cell. Does that answer your question, John?
Yeah, I got it.
Okay, thank you. All right, gang, so now with this new method of sketching things out that I've defined for you on the left side, I was rushing before class to try to get all of this done. Let me come up here and finish a couple little notes. Okay, for the reciprocal crossover here on the right. So for the second question, we're actually going to have the chloroplast genes, which I'm defining as cytoplasmic, being sensitive, and the nuclear genes as being sensitive. And the mating type minus is going to have the chloroplast gene, cytoplasmic, being resistant, and the nuclear gene being resistant. Folks, what I'd like you to do at this time in your own notebook is follow the path that I've outlined on the left, repeat it on the right, and see what results you get. I'll give you a minute or two to do that. As you do that, I will just slowly draw my own very poorly drawn diagram up here, but try to do it on your own. When you've finished your drawing, if you just give me an indication in the chat window. And see if through your drawing you can in fact answer the question, what result would occur in the reciprocal cross? In other words, are all the progeny going to be resistant? Are all the progeny going to be sensitive? Are some of the progeny going to be resistant? Are others going to be sensitive? How would that play out? Go ahead and definitely document that in your notes. Okay, so I'm just going to continue to move us along here, but continue to do your work as you need to do it. And I'm hoping you're coming to the same conclusion that I'm coming to and that I want you to come to, is that in the reciprocal cross, mating type minus carries a cytoplasmic chloroplast resistance gene, which of course is going to break down after fertilization. That means that the cells that are produced after meiosis, they will only carry a sensitive gene in the chloroplast. And then they will either have a sensitive nuclear gene or a resistant nuclear gene, meaning that 50% of the progeny are going to be sensitive to streptomycin, and the other 50% of the progeny are going to be resistant to streptomycin. If memory serves me correct, most of you had indicated that all of the progeny would in fact be sensitive, and that is not true, folks. You can see it here for yourself that only 50% of the progeny would be sensitive. The other 50% of the progeny would in fact be resistant. Okay, so I don't want to beat a dead horse on this one, but I do want to be comfortable. I want to be confident that you're understanding this. So do you have any questions about this? Does the logic of this make sense?
It makes sense, but is there a specific name for this type of like effect, like this degradation of the negative mating type happening like in other species?
In other species? Yes, definitely in other algal species. I'm not aware of a specific example in the animal kingdom. Okay, but that's not to say it doesn't happen in the animal kingdom, John, because you think about it, you know, how many animals do we really study? So, you know, it's possible that it exists out there in other cells and other species that we don't know about it. Other questions, gang? Okay, well, I won't beat a dead horse. I just wanted to point out that, in fact, the answer to the second question here, part B of number 3, was in fact 50% sensitive and 50% resistant. Okay, if there's no other questions on that, then let's talk about recombination. Okay. Recombination is a pretty heavy topic. It's an important topic, but as you can see, there's only so much time we have in the course. That being said, I did want to give you a little bit more exposure to how homologous recombination happens. You know, when we talked about meiosis in the first chapters, in the first third of the course here, we know that crossing over happens in prophase 1 of meiosis 1. We know that, and we stated that, but we never went into the molecular details of how that process was carried out. And so it is in chapter 7 of your textbook that the process is described, but it's pretty intimidating. I mean, it just is. It's kind of intimidating to read the book and understand what the heck is going on here. So I'd like to turn your attention to homologous recombination on figure 18 of your textbook, page 198. Okay, if you have your textbook available, please turn to page 198, figure 18. Okay, folks, as with most things, the only way to understand this is to truly go through it step by step. And there is no shortcut to learning this stuff. Now, that being said, I don't want to take another hour here, but maybe another 20 to 30 minutes with you and try to give you a feel for what figure 18 is trying to actually communicate to you. But I think it will go a very long way if you see a cartoon about this first, because the cartoon will set the backdrop for our conversation about figure 18. So with that being said, I'm going to switch to another YouTube video. It's about 3 minutes long on recombination. Here we go, gang. Just bear with me a moment. Okay. So first off, let's see here. Okay. In your textbook, figure 18, letter G in the textbook, do you see that letter G is labeled holiday structure? Holiday structure. And you notice the title of this video is the holiday junction resolution. So I want you to see that this video is relative, is important for your understanding of figure 18. Okay, enough talking. Let's run through this. The narrator for all of these videos coming from Oxford Academic kind of sounds like he's got a stuffed up nose, and I apologize for that, but that doesn't make the message of the video any less important. So let's watch it and see what this has to say. 1 second here. Okay, just confirming some settings, and here we go.
In a holiday junction, two homologous duplexes are linked by the reciprocal exchange of a pair of DNA strands. The formation of a holiday junction is initiated by the formation of a D-loop between homologous duplexes in which a strand from one duplex, shown here in grey, invades the other duplex, shown in pink. Regions of heteroduplex DNA are formed, in which complementary strands from the two different duplexes are paired. Cleavage of the D-loop and subsequent ligation results in a holiday junction, which links the two duplexes.
Folks, pay attention there. Did you notice that the pink strand literally rotated so that it could base pair with the top strand? And the bottom strand of black here rotated so it could base pair with the bottom strand of the pink strand. Let me back up and watch that again in slow motion.
Holiday junction, which links the two duplexes.
I didn't go back quite far enough. Here we go.
Cleavage of the D-loop and subsequent ligation results in a holiday junction, which links the two duplexes. Holiday junctions adopt a planar, unfolded structure when they interact with recombination proteins. Notice that both of the linked duplexes contain regions of homoduplex, where grey strand pairs with grey strand and pink strand pairs with pink strand, and heteroduplex, where grey strand pairs with pink strand. Also notice that the holiday junction involves 4 DNA strands.
Folks, you can see that on figure 18. Take a look at, pictures F, G, even H a little bit, and the electron micrograph in the center of figure 18. This is what they're talking about, and this is what we're going to be talking about pretty soon.
Also notice that the holiday junction involves 4 DNA strands. There are 2 axes of symmetry in the holiday junction, one horizontal and one vertical. 2 strands of the 4-strand holiday junction are cleaved by symmetrical nicks made by a specialized recombination enzyme called the resolvase. This cleavage can occur across either the horizontal or vertical axis. Note the very different consequences of the 2 cleavages. Upon cleavage along the horizontal axis, the parental segments, big A in grey and big B in grey, and little a pink and little b pink, are still covalently linked to each other. Cleavage along the vertical axis, however, leaves big A grey and little b pink, and little a pink and big B grey covalently linked to each other. Ligation of the cleaved strands generates pairs of progeny duplexes. Holiday junction cleavages along the horizontal axis result in product duplexes in which the parental segments remain covalently linked. They have not been exchanged. These duplexes are called non-recombination products. By contrast, holiday junction cleavages along the vertical axis result in product duplexes in which parental duplexes have been exchanged. In other words, flanking segments from each parent are covalently linked in the product duplexes. For example, big A, in grey, is now joined to little b, in pink. The progeny duplexes are called recombinant products. Both pairs of progeny duplexes also contain regions of heteroduplex DNA in which complementary strands from each parental duplex are paired. Thus, formation and resolution of holiday junctions can result in the permanent exchange of DNA segments between homologous parental duplexes.
Okay, gang. So 3 minutes of pretty intense stuff there. And what I'd like to do is spend maybe 15, 20 minutes walking you through figure 18 to help you understand what this is and hopefully make some better sense of the video. Before I shut the video completely off, do you have any questions?
Yeah, I have a question. What actually like physically makes the strands like open and twist to form the structure to begin with?
Right. There's a whole series of proteins, many of them having enzymatic activity, that some of which I will hint about in the notes I'm going to give you that help make this process happen, Ian. Other questions? Okay. So this is one of those things that, you're looking at your textbook, you're reading it, and that figure 18 looks a little bit scary, so you kind of just ignore it because it is scary. I mean, you really have to sit down and think about this if you really want to understand it. So let's go here to another of my very poor drawings. And hold on a sec. I want to get the chat window back up. Okay. So at this point, you should be seeing another one of my horrible cartoonish drawings that a little bit resembles figure 18. If you don't see another one, if you don't see a PowerPoint drawing with a bunch of DNA helices drawn on them that are red and blue, please tell me at this time. Okay. I'm not hearing anybody complain. So gang, if you want to understand this, and I have to say it that way, because if you don't want to understand it, then there's no point doing this. But if you want to understand this, please copy down using color coding in the pens and pencils you have in front of you in your own notebook. Take a full sheet of notebook paper and copy this down as best you can. And I'll give you a few minutes to it. And I'll type in the window, please let me know when you're done drawing your notes. Yes, you could take a picture of the screen, but that's not going to be where the learning takes place. You got to draw it, gang. Thank you, Sean. We'll get started momentarily, gang. Just waiting on a couple more to finish. Okay, I think we're waiting on one more. It's okay. Take your time. Okay. I think that's everybody. Thanks, folks, for being patient with that. I know this is tedious, but that's where the devil's in the details. That's where the understanding is going to come into play. So what I've done here and what you've now copied into your notes is a slightly simplified form of figure 18 on page 198. And what I'd like to do is just walk you through step by step and give you a couple statements or notes about each thing that hopefully, since you've just seen that video, it will help you connect the dots with some major ideas. Okay, so what you're looking at here is one proposed explanation of how homologous recombination can occur. And I say it that way because, you know, this is not a completely solved research problem. There's still a lot of aspects of this process that are still nebulous and not very well understood. Okay, but let's start with number one here. With number one here, what we have are non-sister chromatids, right? Non-sister chromatids. That's why they're colored differently, red and blue. Non-sister chromatids of homologous chromosomes that are synapsed together at prophase 1. And because they're synapsed, they can begin the process of homologous recombination. Okay, so let's just mention that. What you're looking at there for number one is the synapsis. Oh, excuse me, I gotta change my software here. What you're looking at is the synapsis of non-sister chromatids. Okay, and this whole diagram we're looking at, this whole diagram is a model of homologous recombination. Okay, so as we go so at step number one here, notice we have the non-sister chromatids, right? The red and the blue. And pay attention that as I move my cursor, we have a gene here with two alleles, capital A and little a. We have a different gene here with a capital B allele and a little b allele. It's important that you appreciate that, you know, these letters are the alleles that you're used to thinking about, that we've been talking about all semester long. So we have non-sister chromatids with the corresponding alleles of two different genes here. As we go from number one to number two, notice that the appearance of 2 shows that there's a gap in a segment of each DNA strand for each of the non-sister chromatids. That gap is, in the language of biology, will be considered a single strand nick, a single strand nick. Okay, that would have occurred by an enzyme in the cell called an endonuclease. Okay, so kind of getting to your question, Ian, there's this enzyme, there's actually multiple versions of these enzymes in the cell called endonucleases that have the ability to cut the DNA chains internally. What I mean is not on either end, but inside the chain itself. So that's why you have a gap that's inside each of these chains of DNA. Now that gap, that nicking activity that occurs when you're dealing with meiosis, okay, when you deal with meiosis, that nicking is intentional. It's purposeful. The cell did it on purpose. And it's purposeful because the cell wants to promote crossing over through this process. But there's another possibility to consider. And this is going to be tough to write all the notes I want to give you, but I'll speak it to you. Okay. So this can be done with meiosis where the nicking is purposeful, or it could be done through some unexpected source of DNA damage that the environment has caused to the DNA. And in that case, the damage would have been unintentional. So we could have DNA damage. So whether the nick is intentional through meiosis or whether it's unintentional through DNA damage, it doesn't matter. The cell can still use the same process to either promote crossing over or alternatively to repair the damaged DNA. Unintentional is the last note there that I want to give you. Okay, so there are these enzymes in the cell that during meiosis will intentionally nick the DNA. Next, we go from, please slow me down and pause me if you have questions, because we're already just shy of 3 o'clock. And gang, I know that I personally can't go beyond 3:30 today because of family obligations. So let's see what we can do here. Okay, from step 2 to step 3, you notice that some of the pink strand at step 2 has invaded the blue strand. And you've noticed that some of the blue strand from step 2 has in fact invaded the pink strand. Okay, so this is called, hold on a second here. This is called strand displacement. Between 2 and 3, we have strand displacement. And what we mean by that is that the pink DNA is merging with the blue DNA, and the blue DNA is merging with the pink DNA. Of course, the DNA is not truly color-coded in a real cell, but what we have is the DNA of one non-sister chromatid mixing with the DNA of the other non-sister chromatid. And I'm trying to highlight that to you with that yellow highlight there in my drawing here, which, is kind of like a highlighter that you might use in class. Okay, and note that when that happens, when you displace the strands, some of the blue strand DNA has to twist like it did in the video. You remember I paused it and backed it up where the strand literally has to twist so it can reorient itself to form the hydrogen bonding necessary with the other molecule of DNA. Now that strand displacement happens different ways in prokaryotic and eukaryotic cells. In E. coli, there is an enzyme that is called REC-A. In E. coli, there's an enzyme called REC-A that is responsible for carrying out the strand displacement that you see finished in step 3 of our cartoon drawing. In human cells and other eukaryotic cells, the name of the molecule that carries this out is called RAD51. RAD51. Gang, please hear me on this. I'm doing this because I know you're inquisitive people and you want to understand, but I am not going to beat you up on exam 2 with this topic. I promise I will beat you up about DNA replication, okay? So, getting back to it, the human molecule that supports strand displacement, the human enzyme is called RAD51, and it might be of interest to you, particularly Caitlin, and let's see, is Morgan online? I don't think Morgan's online, is she? Morgan and Hayden are offline. Okay, so Caitlin, you're the gal in the group right now. It's important that you might be of interest to know that RAD51 requires the protein from the gene that gets a lot of attention in the news called BRCA2. So RAD51 requires BRCA2, BRCA2. And that's something that I simply want to highlight, is that in all of our cells, male and female, we have this BRCA2 gene, the BRCA2 gene, which has a normal, healthy, proper function physiologically to cells, which is to help strand displacement. That's its normal job. But when that BRCA2 gene mutates, when you get a BRCA2 mutation, then the strands don't displace properly and the cells cannot complete meiosis properly, or the cells cannot repair their damaged DNA properly. And that obviously can lead to serious consequences for cancer. Typically, we think of as breast cancer most typically. Okay, so BRCA2 is a normal gene that's involved in homologous recombination. When it gets mutated, then we have problems with the recombination and meiosis doing its job properly, which then leads potentially to cancer and other devastating consequences. Okay, questions, gang? All right, so RAD51, along with BRCA2 in our human cells, is helping the strand displacement that you see in step 3. At that point, this branch here, which I'm look at where I'm circling with my cursor. That branch is sort of on the left side of the DNA molecules we've drawn. There's a whole bunch of stuff that happens so that that branch, that opening, now physically migrates to the other side of the molecule. That's why I've said here at number 3 that we have the nick opening and the strand displacement on the left side of the molecule, and at step number 4 that we now have it on the right side of the molecule. So coming from step 3 to step 4, we have a process that is called branch migration. Branch migration. Okay, and what I mean by that is the purpose of the branch migration is simply to increase the length of the exchange DNA between the between the non-sister chromatids of the homologous chromosomes. In other words, what I'm trying to say is branch migration ensures that even more pink DNA goes over to the blue DNA, and even more blue DNA goes over to the pink DNA. My drawing doesn't really accentuate that very well, but that's what I mean by branch migration. Branch migration means we increase the length of the DNA that is swapped between the non-sister chromatids. Increase the length of DNA swapped between non-sister chromatids. Okay, in E. coli, branch migration. In E. coli, branch migration is carried out by 2 proteins, RAV-A and RAV-B. RAV-A and B are responsible in E. coli for the branch migration. In human cells, and generally in eukaryotic cells, The name of the enzyme that's involved in branch migration is called RAD54. RAD54. Okay? The point of me telling you this, gang, is not that I have a bunch of new stuff to make your life difficult on an exam, because I promise I am not going to beat you up with this on the exam. But my point is that you begin to see that there truly are proteins that are involved with these DNA molecules that are physically manipulating and twisting the DNA molecules for intentional purposes. Okay, so between step 3 and step 4, we have migrated the branch. We've increased the length of DNA that's swapped between the non-sister chromatids. At this point, I've taken a little bit of liberty, but what I want you to appreciate is this. Between 4 and 5, let's take a look here. Between 4 and 5, we're going to form the holiday junction here. So number 5 is the holiday junction. I don't think it's anybody's idea of a holiday because it's hard as hell, but it's called the holiday junction. And so, folks, the thing that I need you to appreciate is when you look at number 4, do you notice that little b is opposite on the other side of the molecule as capital A? But down in 5, little b has now been repositioned to be next to capital A. The only way that can happen is if there is a 180-degree rotation of the bottom blue DNA relative to the top red DNA. 180-degree rotation of the blue DNA. Okay. So, there are enzymes, there are proteins here that are facilitating this rotation. I don't have the name of a specific enzyme to tell you, but this doesn't happen magically. There are proteins involved in starting out and controlling this rotation. So just for a moment, I'm going to stop sharing so that you can hopefully see me. And folks, so what I'm saying to you is this, if you can see me, is that if my top hand is the pink DNA and my bottom hand is the blue DNA, notice that my fingertips are on the same side. What's going to happen during strand displacement is that the bottom strand is literally going to rotate 180 degrees in the opposite direction. That will be carried out by these enzymes that are holding the DNA in position. So we go from here to here. There's a physical 180-degree rotation. Okay, back to the PowerPoint presentation. Okay, so at step 5, we have the holiday junction that formed from the DNA at step 4 physically rotating. At this point, you recalled in the video that the video said that we could cut the holiday junction along 2 axes. Okay, and so let me do this. Hold on a second here. Okay. We could cut the DNA this way, right? We could cut it vertically. And if we cut it vertically, okay, so over here we'll say this is the vertical cut. If we cut it vertically, look what's going to happen. Capital A is now physically on the same DNA molecule as little b. But it wasn't when we started, was it? When we started, capital A was on the same physical piece of DNA as capital B. But if we cut the holiday junction after the bottom DNA is rotated relative to the top DNA, then what happens is you separate the left side of the holiday junction from the right side of the holiday junction, and you end up with two physically separate DNA molecules where now capital A is physically attached to little b. And on the other molecule, you have capital B physically attached to little a. In other words, gang, this is what you get with a vertical cut. This is what happens when you have crossing over. Does that make sense? Can you visualize that with this cartoon? How if you cut the holiday junction vertically and took the left side and glued it together and took the right side and glued it together, you'd have the molecules you see in 6A, where now we have the alleles that previously were not connected to each other, all of a sudden connected to each other. That's what crossing over does, and that's basically how we think it is achieved. Okay, so vertical cut. If instead the cells decide to cut the holiday junction on the horizontal axis, we have the horizontal cut leading to 6B. Okay, and at 6B, you take the two top sections and lift them out, and the two bottom sections and lift them out. And what you can see is that the allele positions are preserved. We have capital A next to capital B, which is the same thing we started with at step 1. And on the bottom strand, we have little b next to little a, which is the same thing we started at with step 1. So with the horizontal cut, there is no crossover. We're not exchanging the alleles. There's no crossover. But a cell might still want to do the horizontal cut because it is trying to repair the damaged DNA where you see the nick in step 2. So the horizontal cut would be used to repair damaged DNA. Okay. So the vertical cut and the crossing over is what you're going to expect a normal, healthy meiosis to achieve. Whereas a horizontal cut is what you're going to expect when the cell is not interested in meiosis, but it is interested in DNA repair. Okay, gang, that is my spiel. You have thoughts or questions?
I have a two-part question here for you. So specific to crossing over, I know we discussed this previously, but this last hour made me appreciate the complexity a little bit more. How likely or how frequently do errors occur here? And then what would be the associated implications of that?
Well, let me think about that for a second. I think, to answer your question best, usually when this process goes haywire, down the road, it does lead to some level of tumor formation, which then may progress into cancer formation. So I don't have a number that I can quote you off the top of my head, Kevin, but I think if you took a look at epidemiological statistics of how frequently breast cancer shows up in our population, whether it shows up in, one person in every 1 million or whatever it is, I think that would be a fairly rough analogous number that would tell you how frequently this process messes up. What was the second part of your question?
I think you answered it all in one there. I was inquiring about the implications of that. So are we presuming that this here is where the cause of breast cancer really lies, or is it really just theory?
No, it's not so much theory. And, it's easy to poo-poo theory because it's theory and it may not be reality. But theory is the only thing we really have to tangibly work on in science until we figure out something that is more accurate. But we definitely know in breast cancer and other similar cancers, we definitely know there is mutated BRCA2 proteins that are mutated because something's happened to the BRCA2 gene. And because that protein's mutated, strand displacement doesn't function properly, crossing over doesn't work properly, and the chromosomes kind of get, how should I say it? They kind of get intertangled in each other, and they can't separate properly during meiosis. So that leads problems with polyploidy and unhealthy numbers of chromosomes, which then promote deranged cell behavior, which certainly includes cancer. So it's not fantasy land. We really do have scientific data to support the theories that are being proposed here. Any other questions?
So I'm a little confused about the horizontal cut and the DNA repair, like that type of this happening. When would that occur? Because this is all happening during meiosis, right? Like this
Okay.
can't occur like not in meiosis.
Well, remember, John, what we're talking about is homologous recombination. That is used by meiosis, but it's also used at other times the cells are not performing meiosis and simply living their lives. So what I'm saying is, what we're talking about is homologous recombination that uses meiosis, or suffice it to say that I should say it this way maybe. Meiosis sometimes uses homologous recombination, but even when cells aren't performing meiosis, they may still perform homologous recombination to repair damaged DNA. So you asked, when could this happen that doesn't involve meiosis? Well, tell you what, the next day that's nice out, 60 or 70 degrees, go outside, get some sunshine on your skin. Undoubtedly, there will be a mutation in a skin cell somewhere on your body, and your body will use this process to repair the damaged DNA in your skin cells. And if it does a good job of it, you're not going to develop cancer. You're not going to develop skin cancer. If it does a bad job of it, which I hope it doesn't do that, then maybe 15 years from now, maybe 5 years from now, you develop a patch of skin cancer on your body. Does that make sense?
Yeah, that makes sense.
Okay. Gang, what else? Okay. I think we've maybe done you in at this point. So, okay. All right, gang. So do you have any questions about any of our conversation today before we sign off? Looks like Caitlin needed some comfort from her cat. Okay. All right. Okay, gang. So here's what to expect from me. At some point late tonight or early tomorrow morning, I'll get you an email out with a recorded link for today's lecture and perhaps some other videos. You'll get that information from me before the weekend. And because not everybody could be in lecture and not everybody's here in our time together in the office hour, I will put together a genetic study guide for lecture exam 2 and give you some hints about things that you need to prepare for your exam in 2 weeks. Do you have any thoughts or questions?
Yeah, I have a question. So now that we know we aren't going to be going back to the campus this semester, what's going to happen with like labs?
Yeah. At this time, Ian, my feeling on labs is that most of the labs I had intended to do with you, there's no way to make these things up virtually. So most of the labs, you're simply going to be awarded credit, even though we actually haven't done it. I think that's the fairest thing I can do is, you know, it's nobody's fault. It's not your fault. It's not my fault. We just can't do this. There is one lab that I'm still thinking about that, you know, you might be able to do at home on your own time, I'm thinking about possibly assigning another lab as a homework assignment. But I will let you know and give you plenty of warning on that if I decide to go that route. So the short answer is, for right now, labs are done for the semester, and for the most part, you're going to be given credit for them.
All right, thank you.
Anything else?
Regarding the question we discussed earlier. So that ties into what I asked previously, but is it safe to assume a lot of what we discussed was specific to prokaryotic transcription, or are there some parallels between prokaryotes and eukaryotes?
Okay. We were specifically talking about prokaryotic transcription today, but there are a lot of parallels. There's more parallels at the undergraduate level that I would want you to appreciate than there are differences that I would want you to worry about. Okay?
Thank you.
All right, gang. Anything else? All right. So stay safe out there. Try to enjoy your weekend, and we'll be back together next Tuesday. I'll see you later.